This video is prepared in the partial fulfillment of the requirement towards the project of course SC462 Elements of Synthetic Biology Life 2.0 Spring 2016. Professor Manish K. Gupta is the instructor of this course. My name is Shreyansh Prajapati and I am a BTech third year student. My ID number is 2013-01062. And in this video presentation I am going to talk about CRISPR and HIV. This project is done by two members, Shreyansh and Dilip, in which we have reviewed research papers and articles related to CRISPR and HIV. We have submitted the report containing our understanding of the problem domain. The purpose of this project was to understand and look into the details of effect of CRISPR on HIV virus. In this video, I am going to cover CRISPR, its application, HIV, stages of HIV, use of CRISPR-Cas9 with HIV, test conducted and its results, observations, how HIV beats CRISPR and the future scope of this project. So let's start. Over the last few years, scientists have developed various genome editing methods such as zinc finger nuclease (ZFN) or transcription activator like effector nuclease (TALEN). This molecularly programmed nuclease can identify and cut at a specific piece of nucleotide sequence of targeted genome sequences. These technologies enabled us for transgenetic mutations such as substitution, deletion, and insertion induced by host DNA repair machinery. ZFN or TALENs are somewhat difficult and time-consuming to develop, design, and empirically test in a cellular context. These drawbacks resulted into development of new technology called CRISPR. The full form of CRISPR is clustered regularly interspaced palindromic repeats. So the question arises is, what is CRISPR? CRISPR is a natural defense mechanism found in the bacteria. CRISPR-Cas9 is a genetic engineering technique that makes use of this cellular machinery that bacteria use to identify and modify the DNA of harmful viruses. Clustered regularly interspaced short palindromic repeats are segments of prokaryotic DNA containing short repetitions of base sequences. Each repetition is followed by short segments of spacer DNA from previous exposures to a bacterial virus or plasmid. CRISPR is one part of bacteria's immune system which keeps beat of dangerous viruses around, so it can recognize and defend against those viruses next time they attack. In the simple words, we can say that CRISPR is a genetic engineering technique in which DNA can be inserted, replaced, or removed from a genome using artificially engineered nucleases or molecular scissors. This technique has two major components. The first one is Cas9. Cas9 is an enzyme that can cut a double-stranded DNA at a very specific block, its gene expression. In CRISPR, gRNA is a short strand of RNA called messenger RNA. CRISPR will guide the Cas9 enzyme to bind with a specific sequence on a genome where Cas9 can make a specific cut. So the task of the Cas9 is to cut at a specific location in the gene. CRISPR has many applications in very different areas. Using CRISPR-Cas9 technology, one can insert, delete, and modify DNA in human genome and animal. CRISPR can be used in plants also. There are many applications. Some of them are listed over here. CRISPR-Cas9 technique can be used to cure a rare liver diseases, cancer, and HIV. CRISPR-Cas9 technique is used to eliminate muscular dystrophy in mice. CRISPR is used to create transgenic animals such as pigs, zebrafish, rat, and mice. Food manufacturing companies are using this technique to produce cheese and yogurt. Using this technology, we can improve the quality and life of the rice, tomato, tobacco, etc. CRISPR is also used to cure blind eyes. Now we are going to now we are going to cover HIV in much more detail. Our immune system defenses our body against the external host such as viruses. HIV is the virus whose target is human immune system itself, made it known as human immunodeficiency virus. White blood cells plays an important role of immune system. HIV destroys certain white blood cells called T helper cells or CD4 plus cells. If too many of these cells are destroyed, then body will no longer be able to defend itself against the infection. If at the earliest stage HIV is not treated and identified, then it reaches its last stage known as AIDS. Time spent time period to reach AIDS is usually 10 to 12 years. How much quickly virus develops in a body depends on the various factors like diagnosis, overall health of your body, and how well the treatment is taken. But we can cure HIV with CRISPR-Cas9 genome editing tool. For that, now we are going to understand four different stages of HIV. The first stage is a binding and fusion. In the first stage, HIV virus spikes help it to join with T helper cells. This, as we can see in the picture, these are the spikes and this is the T helper cell. So using these spikes, HIV virus can attach itself with the T helper cell. We will see this in the next picture. Second stage of the HIV is called reverse transcription and integration. Part of the body HIV starts translating HIV RNA into HIV DNA using an enzyme called reverse transcriptase. As we can see, the spikes are connected with the T helper cell. Now transcription of HIV RNA into HIV DNA is happened in this stage. Third stage of the HIV is called transcription and translation. To produce more HIV DNA then makes long strands of messenger RNA proteins and transport them toward the edge of the cell. So in the fourth stage, named as assembly budding and maturation, new HIV particles are formed and released into the system to spread infection all over the body. This is how the HIV viruses spread into our entire human body. Now we are going to talk about the test that conducted by the researchers, in which green fluorescent protein is inserted into HIV DNA to measure viral gene expression. 80 PAM sequences were identified. 10 gRNA sequences ranges from T1 to T10 are selected by blasting of human genome. 4 of them 10 target sites are among these 10 target sites. 3 are in LTR, 5 in pole gene, and 2 in second exon of TET. Then gRNA are nucleotransfected together with Cas9 plasmid DNA into JLET 10.6 cell. The targeted viral DNA regions were also amplified by PCR and further examined by the sequences. Though the score of the T10 guide RNA was very low, it caused the most type of deletion and insertion into the HIV virus. So how did CRISPR help? CRISPR, being a genome editing tool, can easily cut out the infected DNA containing HIV DNA and can easily remove it and completely remove it from our body. Paper is discussed our report which demonstrate the effectiveness of our gene editing system in eliminating HIV from the DNA of T cells and by introducing mutation into the viral genome, permanently inactivating and activating its replication. Recent results says just a few days back, there is a statement made by Dr. Chen Ling. It's like a leftover HIV puts on a fake mustache before going back to work ravaging your immune system. Meaning, when the CRISPR edits or cleaves the HIV genetic sequence, the remaining or leftover virus mutates enough so that HIV CRISPR do not recognize. So HIV viruses are very smart. They know that CRISPR is editing or cutting, so they mutate itself so that CRISPR won't be able to recognize where to cut. Recent studies show that HIV does beat the CRISPR in the first round, but still we have hope as new methods can be derived so that HIV can be completely cut out from a DNA without leaving any leftovers. Because if there is any leftovers, if any part of the HIV is remained in our body, then it will replicate itself, so that HIV cannot get a chance to regenerate. Yes, HIV does beat CRISPR in first round, but still this is not the end. There is a lot more work to be done in getting this technique ready for something more advanced than just putting out human cells in a petri dish. According to me, it is also possible that HIV particle itself can be hijacked to cure HIV, as their design already permits them to target specifically CD4 plus cells, which can be used as a delivery method. So, if the if we can use HIV virus itself to hijack HIV to cure HIV, CRISPR can easily find the HIV virus in the human body. These are some of the references that we have used. You can also refer to our report. Thank you.